This presentation will provide an overview of how whole genome sequencing can be used to identify Salmonella virulence factors, to answer the question as to what data we do have right now to inform Salmonella priority serotypes for intervention. Here the keys is a summary and the key conclusions from this presentation. Number one, Salmonella shows tremendous diversity. It's a large number of different serotypes with different virulence genes, etc. However, only a small number of serotypes are responsible for most of the human salmonellosis cases, such as enteritis and typhimurium, while other serotypes that can be frequently found in some sources, such as poultry or meat or beef, are unlikely to cause human disease and also appear to show attenuated human virulence, so reduced ability to cause human disease. The key take-home message from this is that a targeted control of serotypes of human public health relevance is a promising approach to reduce the public health impact of salmonellosis. One practical way of how this could be implemented is to take serotypes of limited public health relevance, such as Sero in Kentucky in the US, and underweigh them in the performance standards to allow for more effective pre-harvest control of those Serovars that are of human public health relevance. Whole genome sequencing can help inform these definitions of what we'll call hypo and hypervirulent Serovars and clades. However, that's not going to be simple. There isn't going to be just one marker that tells you hypo or hypervirulent, but it's going to be more likely to be a convergence of whole genome sequence data, epidemiological data, and phenotypic data that will identify those Salmonella of high public health relevance and those Salmonella of limited public health relevance. So what I'm going to go through today to get you those conclusions is a little bit about Salmonella, just to get us all started. Salmonella represents two species. The whole genus is considered a pathogen, and over 2,600 serotypes that are organized in a number of different subspecies of Salmonella enterica, as shown in this picture. Salmonella enterica, subspecies enterica, is the major subspecies of public health relevance. That includes the largest number of recognized Serovars. Salmonella is one of the most important global foodborne pathogens, as seen on this picture on the right side, with both Salmonella typhi and non-typhoidal Salmonella, causing the largest numbers of disability-adjusted life year losses globally. And meat and poultry is a major source of human salmonellosis infections. Now, having said that, we realize now that Salmonella is not just transmitted through direct animal contact. It's not just our classical zoonotic pathogen. Environmental sources, persistence of Salmonella in processing plant, particularly those making dry products, cereal, bakery products, chocolate, is also an important source of salmonellosis. But the big one still is meat and poultry. Even though Salmonella can be the same subspecies, so enterica, subspecies enterica, different Serovars, different strains can cause vastly different diseases. And the textbook example for this is Salmonella typhi, human-specific infection, very severe symptoms, invasive disease, no gastrointestinal symptoms, versus the rest of the Salmonella that we call non-typhoidal Salmonella. Shorter incubation period, this is your classical gastroenteritis that is usually self-limiting, but in some Serovars, more frequent in others, causes also invasive disease and more severe symptoms, including possibly some long-term sequelae. But even within those non-typhoidal Salmonella, we have a wide variation of symptoms, severity of disease caused, host specificity, etc. One of the key ways we organize Salmonella is Serovars or serotypes. These are typically used interchangeably. Serovars are based on surface molecules in Listeria, and specifically on three surface molecules: the O antigen, which is the lipopolysaccharides, and the flagella antigen, which come as phase 1 and phase 2 flagella antigens. Each Serovar really can be designated, typically designated with two different ways. One of them is a name, such as Mississippi, or typhimurium, or enteritis, and with a numerical pattern. where the first numbers here before the semicolon are the O antigen, the middle one is the flagella 1, and the last one after the last semicolon is phase 2 flagella. Some Serovars only have a numerical profile, and probably the most well-known is 4512I minus. This is known also as non-motile or monophasic Salmonella typhimurium. The minus indicates that the phase 2 flagella are missing in this strain. Salmonella typhimurium will be 4512I12. And this monophasic typhimurium, as well as some other ones, actually don't have a corresponding name to it. These are only referred to as 4512I minus. Now, with serotypes and solely relying on serotypes to characterize Salmonella and identify those that are more or less of a public health hazard is non-trivial, because many serotypes are what we call polyphyletic. Even though they're the same serotype, they represent two completely distinct groups of organisms. As shown here in Mississippi, Mississippi has two clades, clade A and clade B, or at least two clades. Clade B is found here in this giant tree of different Salmonella serotypes. Clade A is found here. So even though they're both Mississippi, they both have the same surface molecules, they're very different and they're actually found in different continents. The same thing can happen that you have two different clades that cause different types of disease. So we can't always, we shouldn't rely on Serovars alone to classify Salmonella because of this. The second issue is that even if a Serovar is monophyletic, or even if it's a monophyletic clade of a Serovar, within that Serovar, you can have very distinct subgroups. This is an example of Salmonella sero. This tree now is all the same Serovar, but what you can see is that some group here, all of the isolates here in this tree have lost SOP-A, a key virulence gene. Another subgroup here that comes from this ancestor lost Phys-A, another virulence gene. So even within a Serovar, we can have distinct subgroups that differ in their likelihood to cause human disease. So take-home message, can't use Serovars alone to say this Salmonella is of particular public health relevance and this one is of less public health relevance. We need to take whole genome sequencing information, other information to make those decisions. So what Salmonella virulence genes are out there? How can we use Salmonella virulence genes to inform these decisions as to which Salmonella subtypes or Serovars of public health relevance and which one of less public health relevance? Salmonella virulence, it's complicated. Salmonella isn't just, an E. coli or Listeria that goes by another name. With sugar toxin producing E. coli or enterohemorrhagic E. coli, it's difficult, but it's nothing like Salmonella. With STEX, we have a specific set of toxin and virulence genes, STIX1, STIX2, EAEA, and they can help and can be used to define whether an E. coli is likely to cause enterohemorrhagic disease. With Salmonella, we have 24 currently known Salmonella pathogenicity islands, known as SPIs. And each pathogenicity island has multiple, typically multiple, and a number of different virulence genes. So with this tremendous diversity, there's no reliable or agreed upon way to take, Salmonella virulence gene presence absence data from, let's say, whole genome sequencing and say, this Salmonella is going to cause human disease. This one is not going to cause human disease. It's a lot more complicated than that. So we don't have an easy prediction based on virulence genes. This is just to give you some idea how complicated it is. These are only 3 of these Salmonella pathogenicity islands: Salmonella pathogenicity island 1, Salmonella pathogenicity island 2. Here are the different genes within Salmonella pathogenicity island 1. Here are different Salmonella Serovars. Green shows that the virulence genes is there. Black shows it's not there. And so you can see many of these virulence genes are found essentially in all Salmonella. Some of them have presence absence patterns. And this is only 2 or let's say 3 of these 24 SPIs. So clearly very complicated. We have a way to track virulence factors, or I should say, we probably will have a way to track virulence factors based on whole genome sequencing through the pathogen detection platform in NCBI. If you go to today for Salmonella, it will actually give you virulence genes. But to the best of our knowledge, when we looked at this, it doesn't seem to be the actual Salmonella virulence genes. It actually seems to be the E. coli virulence genes. So what we take from this is that NCBI is working towards this, but definitely don't at this point try to use this to make any predictions based on virulence. You need to do this with individually with someone or a group that knows Salmonella virulence genes to use individual whole genome sequence their data to predict which virulence genes are present. So how do we use this information on diversity and virulence differences? First, let's talk a little bit about sort of global diversity. So we can see these are the top 5 Serovars in different countries, regions of the US, regions of the world. And you can see, a couple of things that Typhimurium and Enteritidis are high up globally. These are some of the global high priority Serovars. But then some of the other ones will differ. We have Javiana as number 4 in the US, is also high as number 5 in Argentina. But then in some other countries, you have completely different Serovars high up. So in Thailand, for example, Valtverraden, Riston, and Stanley are high up. And Valtverraden in particular tends to be one that I at least associate more with Asia, et cetera. So tremendous diversity globally. These are the top Serovars in the US, Enteritidis being number 1, causing 20% of human disease cases, roughly Newport, Typhimurium. And so you can see the top 5 serotypes alone cause more than 50% of human infections. That's 5 out of 2,600, which gets me back to this and our initial hypothesis is that if we focus on the top Serovars, that's one way to make continuous improvement, a faster continuous improvement in terms of reducing human Salmonella infections. The serotype profiles also differ by sources. Again, these are US data. You have Serovars common among human disease cases. And then you can see among egg products, Enteritidis is high. Among ground turkey, other types of Serovars are high. Among chicken, broiler chicken, Kentucky and Enteritidis is high. If you look at dairy operations, we're going to come back to Salmonella sero is high. You can see the most common serotype there, but doesn't show up in the top human Serovars. Broiler chicken, Kentucky high up, doesn't show up in the top human Serovars. So these are 2 strains that cause us headaches and I think ultimately distract from reducing human salmonellosis. If we have performance standards, such as we do have for broiler chickens, where we say we have to stay below a certain percentage of chicken positive. If we want to reduce that number, Kentucky is a good target because it's one of the most common Serovars. If we reduce Kentucky or similar use sero in dairy operations pre-harvest, we might reduce the prevalence of Salmonella. We have less Salmonella there, but we're not going to have a public health impact because these Serovars, at least in the US, are not an issue with human disease cases. And likely there are other Serovars with similar situations. So the hypothesis, again, to be more specific, is let's not focus on Kentucky and sero. Let's design systems that these ones are not weighed highly in, for example, these performance standards, so that we focus on the other serotypes that are important, Enteritidis, Typhimurium, and Phantis 4,5,12 All of these show up in the top human cases. Sero doesn't, Montevideo, Newport, and München do. So for both dairy and broiler, the top Serovars are really essentially distractions for public health impact. So that brings us to the idea that serotype-specific interventions at source show potential for public health impacts and improve public health impacts. Why do we need to be serotype-specific? People will say, well, Why do we need serotype specific? Let's just put interventions in case that work for all Salmonella. Vaccinations have to be serotype specific. Most vaccine at the best, you can vaccinate against 2 to 3 different serotypes. So we need to define those Serovars of greatest public health importance. So what do we propose? We propose a multi-pronged approach that classifies Serovars as more or less likely to cause human disease based on epidemiological evidence, what I've showed you before, relative frequency in animal source populations and humans, presence, absence of virulence-associated genes, and phenotypic evidence that certain strains may be less likely to cause human disease. If we synthesize all of these data together, we should be able to have reliable information as to which Serovars, which groups may have reduced and will have a reduced likelihood to cause human disease, and therefore should not be targeted in pre-harvest intervention strategies. Let's make it tangible. Salmonella sero as an example. We talked about it before, counts for a very small number of human illness cases, only 38 confirmed human illnesses in 2016, despite being the most frequent serotype among dairy cattle. And dairy cattle are obviously an important source of human food, particularly ground beef. But that's not just it. We find that a number of virulence genes are absent or have mutations that make them non-functional, such as SOP-A, FIS-A, etc. When we test Salmonella sero in tissue culture, we try to infect human cells, they show lower invasion efficiency. So this is what I call this classic convergence of epidemiology, phenotypic, and genomic data that support that sero is reduced public health issue. How do we do this on a larger scale? We can mine data, such as shown here, where we can mine the frequency of these different strains among non-human, non-clinical isolates, compared to the proportion among human clinical isolates. And you can see that certain of the high priority serotypes, Newport, Typhimurium, and Enteritidis, fall along above this middle line. What does that mean? Even though there are fairly few non-human, non-clinical isolates of Newport, it's much more frequent among human cases. So these are the ones that are definitely of high public health relevance. On the other ones, the strains here, like Kentucky, very frequent in the databases among non-human, non-clinical isolates, but very infrequent among human cases. Similar Senftenberg and Bedonka and Sero. So this will be the epidemiological evidence as detailed before. This should be supported by other evidence, such as phenotypic data and WGS data, but with that we can define Serovars of reduced public health relevance. And as a reminder, this should be country or region specific. Yeah, just because Kentucky is of reduced public health relevance in the US doesn't mean that's global. And then we can use that to really target interventions towards the Serovars of public health relevance. How would that work? If you take a hypothetical poultry flock right now, where you may have different Serovars at a different prevalence, and typically Kentucky will be the most common, then Typhimurium, then Berta, then Newport, then Enteritidis. If I simply want to get the numbers down, and I can only vaccinate against 3, I will probably vaccinate against the top 3, Kentucky, Typhimurium, and Berta. If we were to say we have evidence that Kentucky is unlikely to cause human disease, therefore we introduce an adjustment factor to underweigh this by, let's say, a factor of 1/10, saying even though it's 10%, but for public health adjusted relevance, it's only 1%. Now our top Serovars become Typhimurium, Berta, and Newport. Newport, very common among human disease cases, even sometimes an issue with multi-drug resistance. Now we would target our vaccination against the Serovars. Much likely to have a bigger public health impact than this type of vaccination of a poultry flock. So that brings us back to the conclusion. I've introduced you to the tremendous diversity of Salmonella. I hope I've given you some examples how we have a targeted control of serotypes of human public health relevance as a promising approach to reduce the impact of salmonellosis. and given you very specific examples of how we could start out with serotypes that are of limited public health relevance, such as Sero and Kentucky, underweight them in performance standards to allow and encourage more effective pre-harvest control, for example, through vaccines. And then I've shown you how we can use WGS data, but I've given you also some of the caveats of how challenging that might be in Salmonella, which is why we propose that it's not WGS data alone, it's that conversion of WGS, epidemiology, and phenotypic data that should be used to identify Salmonella of high or limited public health relevance. Thank you very much.